Hello everyone, a very good morning. I am Dr. Shweta and I'm here today to give a brief topic about data analytics in pharma. So, I hold an experience of 3+ years in this pharma industry, wherein I have worked on multiple projects related to the domain. So I'll be able to provide you appropriate insights on how you can use your pharma knowledge into the field of data analytics and data science. So today we are going to cover the introduction to pharmaceutical industry, the pharmaceutical industry in India, application of data analytics in pharma, and agenda for the course. Now, what is pharmaceutical industry? Okay. The pharmaceutical industry develops, produces, and markets drugs or pharmaceuticals. Why are we developing these drugs? These are to be used as medications, wherein we aim to cure, vaccinate, or alleviate or reduce the symptoms in patients. It encompasses both the generic and brand medications along with medical devices. Now, what are generic medicines? So there are two things in pharmaceutical industry, generics and brands. Generic medicines are the molecules which are directly put out into the market, whereas brands are the ones who discover it and then market it. Okay. The pharmaceutical sector is known for its significant investment in research and development. With the development of a new drug costing of an average of $2.6 billion. So overall, when a drug is being discovered, produced, developed, all this takes around $2.6 billion of investments, and a huge amount of time is also spent on this. Once a drug is discovered, after a huge research, once it is discovered and it is developed, it overall takes around a decade. That is around 10 to 12 years just to put it out in the market. This also involves huge costs and a lot of unsuccessful trials. Sometimes it happens that once all this much amount of time and this much amount of money is spent, the drug might still be unsuccessful. So the development of drug takes a lot of toll, especially in terms of investments and time. So we are here trying to use our knowledge in data science and data analytics and apply it to pharma so that we can reduce this huge burden. Highly regulated globally by agencies like the FDA and EMA, the industry must adhere to strict safety, efficacy, and marketing regulations to ensure the products are approved for the market entry. Even the globally regulated agencies like FDA, Food and Drug Administration, and EMA, European Medical Agencies, all these have to adhere strictly to the safety and efficacy and marketing regulations so that their product is out in the market. Now, as a $1,000,000,000,000 global industry, it still faces challenges in making drugs accessible and affordable, especially in developing countries. So, These challenges are necessitating us to take initiatives to improve global health equity. Now, coming to pharmaceutical industry in India. So, India is responsible for approximately 20% of the world's supply of generic drugs. So overall, 20% of the drugs, mainly the generics, are being supplied by the India. So it is a crucial player in the global pharmaceutical sector. Okay. India is home to around 3,000 pharma companies, and it has around 10,500 manufacturing units. So this is how the pharmaceutical sector is spread over in India. India has an extensive domestic market. So India's pharmaceutical value was at around $42 billion in 2021. And it is expected to reach $65 billion by 2024. So we can understand how significant the growth is and how much potential is there for pharma industry to grow in India. Now, each pharmaceutical firm spends around 8 to 13% of their revenues on research and development. Okay, so 8 to 13% is still a significant amount, and each firm spends so much only for research and development. As research and development is a very important part and leads to a lot of innovations. This figure keeps growing as companies are shifting more towards innovative products. Coming to regulatory environment, compliance with international standards is critical for access to global markets. Now, Indian companies hold a significant number of ANDAs. ANDAs are nothing but abbreviated new drug applications. So the companies can apply for ANDAs with the US FDA and get the approval to manufacture the generics of a drug. So Indian companies hold a significant number of ANDAs and have around 300 approvals from the US FDA just in 2020 alone. Number has increased from then on. So overall, pharma is a huge market, and in that, India holds around 20% of world supply. So this domain is growing and has the highest potential to inculcate innovations and also analytics. Now, where all can we integrate the data science, data analytics in the field of pharma? To understand that, let us go through the process of how a drug comes into the market. Now, when there is a drug being discovered, first they go for chemical compounds. Okay, these chemicals say this is one structure. Now, a lot of research and development goes into this stage wherein the companies try to find what is the appropriate molecule which will be effective in the treatment of a particular disease. Now, they take this molecule, test it in the labs, see if that particular molecule is affecting the target. Okay, like say if it is a diabetes medicine. Okay, so a particular molecule, say metformin, if this was being studied or researched, they'll see if this metformin is able to go and adhere to the cells which cure diabetes or which will help produce insulin. So this target discovery and this attachment of the molecule to the target is uh, extensively studied. So like this, there might be thousands of molecules which will be studied. Each molecule, they might differ in one or two uh, atoms or very slight changes in the molecule, but the whole theory changes. The way they act on the target changes. So all these thousands of molecules will be studied on the target, and the molecule which is giving better efficacy will be chosen. So this extensive study on thousands of molecules, manufacturing them and then uh, studying them will take a lot of investment time. Okay, researching about this it takes a lot. Once this is done, once they know that okay, this drug is better, then this drug is going for development. Okay. Here it is produced. All the pharmacokinetic and pharmacodynamic factors will be studied. Okay, how the drug will be absorbed in the body, metabolized in the body, digested, excreted, all that. And if you're giving 100% of the drug, how much percent is being absorbed, how much efficient it is, all these will be studied. These all will be studied again in clinical trials. Okay, in this clinical trials, first the drugs are administered to the animals in the laboratory conditions, and then it is tested on humans. In humans also it is tested again on different phases, and once we find out that there are no side effects or no toxic effects, then the drug is released into the market. This whole development cycle takes around 10 to 12 years and billions of rupees. To reduce this, we are trying to use a lot of analytics in each phase. Say, first stage was R&D, that is research and development. In this phase, where we are taking thousands of molecules, in here, we can apply analytics so that we can understand which molecule in the past had given better efficiency and directly take that and study it. So this will reduce a lot of time and effort. Okay, instead of taking thousands of drugs, you can just study on few. So this is one way where we can apply analytics. Next, in the clinical trials, we can apply analytics wherein the people or the patients who are enrolling for the trials, we can apply analytics and select the most appropriate person. Okay. So the most appropriate person will be selected for the trial, and this in turn increases the trial efficiency. So we will get the appropriate data whether the drug is affecting or drug is able to cure the disease or alleviate the symptoms. Alleviate is nothing but reduce the symptoms or about the side effects. So choosing the appropriate candidate for the clinical trials is also very important, and we can apply our analytics over here too. Next, moving ahead, once the drug is released in the market, okay, post-marketing surveillance is done. That is, though the tablet is released in the market, it will still be monitored by the drug companies, and any person who has a side effect will report to the particular physician or the company, and the company collects all this data and tries to analyze what is the percentage of side effect they're facing and how many people are facing, whether they have to change the chemical formula, or they try to see what are the steps taken to reduce these side effects. So in many stages, these analytics can be applied, and in pharma industry, analytics play a very important and a major role. And there is also a lot of scope for improvement, which in turn affects the life of a patient. Okay. Ultimately, we are improving the quality of life. So this was a brief intro, like how we can apply uh, analytics in pharmaceutical industry. Okay. Next, moving ahead. India has a very huge domestic market too, and also it exports a lot of drugs. Now, pharmaceutical exports from India stood around $24.4 billion. Okay, so huge amount of drugs from India are exported. Only in the year 2021 to 22, there was $24.44 billion exports from India. So this is another huge market where we can produce a lot of change. Challenges and opportunities. Despite the huge number, okay, despite there is a huge challenge, the Indian pharmaceutical sector is witnessing a rapid expansion in the contract research and manufacturing services. This contract research and manufacturing services is expected to grow at a rate of 12 to 14% of the next 5 years. Okay. What is contract research and manufacturing services? Now, when it comes to drug discovery, where a lot of research is done and lot of time and money is spent. Okay, so if a pharmaceutical company has to produce a new drug, so they have to invest around 10 to 12 years and also a lot of billions of dollars, so as to get one new product. Because this is time consuming and all, this particular thing is outsourced. So that is nothing but contract research and manufacturing services, wherein as some other company is given or so here a pharmaceutical company is giving or outsourcing their research to another company. So this particular organization will try to discover the drug and also help in the manufacturing. So by doing this, a lot of challenges have been addressed, and now the growth rate is expected to be 12 to 14% in the next 5 years. In the times of COVID-19 also, India has played a significant role. Globally, every country was fighting with the COVID-19 and had to produce vaccines. But India, because of its huge market in pharmaceutical area, was able to export millions of vaccines doses to around 150 countries. So the Indian pharmaceutical sector is leading overall. Now, we exported around vaccines for 150 countries under the Vaccine Maitri initiative. And after this, the Indian government increased the expenditure on healthcare. Okay. Before, it was around 1.8% of GDP. Now it has, before it was 1.3%, and now it has increased to 1.8%. This tells us that there is a significant commitment in strengthening the Indian healthcare system also. So even the government is trying to increase or strengthen the Indian healthcare system. Overall, in the coming few years, pharma industry in India is known to grow exponentially, and integrating analytics into this will also help it in growing and also improving the patient care. There is even digital transformation seen in the pharma sector in India, wherein the adoption of digital technologies is growing, but the digital health market in India expected to reach around $5.43 billion by 2025. This is driven by telemedicine, e-pharmacies, and digital consultations. So there is even a digital transformation in pharmaceutical companies. Now, so the size of the pharma analytics in India is 2 billion US dollars. Okay. Now, this was $2 billion, and now it was expected to grow by 11.5 billion US dollars. Okay. This is the stats until 2020. And further, it is going to grow more and more. Okay. So the growth in the pharma industry is being exponential, and the pharma analytics market size is around $1.5 billion in terms of annual revenue. So these are some of the big numbers. Pharma companies are the big players in even globally and in India. Pharma analytics enables evidence-based rather than intuition-based decision making. So if you are applying analytics in pharma, it is more like taking an evidence-based decision rather than intuition. Like If you're trying to produce paracetamol, paracetamol, as everybody knows, it is taken for fever or it's like an analgesic. So instead of just going into intuition based and telling that, okay, the next season there will be a lot of paracetamol drugs need or drugs being sold. Instead of taking an intuition based decisions, if we apply analytics, then we can understand when this paracetamol is sold more. Mainly, paracetamol is again used for fever or pain relief, and sometimes, say, the seasonal flu. Some seasons have a lot of people getting fever and all. Then you can predict when would be the highest need for paracetamol and stock those tablets accordingly. So by this, analyzing the past data and telling that in Jan to Feb, there might be a lot of need. By analyzing the past data, you can uh, you can tell the pharma companies to produce or manufacture more of these drugs and stock it more during that particular season. So this is how you can go for evidence based rather than intuition based in decision making. With the help of advanced analytics, businesses can identify future markets and avenues for greenfield ventures. So you can use analytics to help businesses like pharmaceutical companies identify which drugs are going to be needed more by which countries and how much to be stocked and all that. You can analyze the future markets and tell the companies, and also there might be avenues for greenfield ventures. You can help them grow in other countries by just analyzing the data. Now, statisticians can develop strategic pricing models to boost revenue growth. We can work on developing various pricing models so that will help in the revenue. The recent talk in pharma sector, which is very loud now. You can hear pharma everywhere, and how analytics is being used in pharma is right now booming. This has led to a surge in the requirement for pharma analytics course professionals in our country. Because pharma is growing and also the analytics field is growing, combining these two is has become the talk of the town. And this has led to a lot of people taking pharma analytics course. Even the pharma companies are moving up the value chain with the help of savvy analytics professionals. Because these people are predicting, are helping pharma companies take decisions. Even most of the pharma companies are depending on these analytics professionals. And this dependency is growing exponentially again. Now, McKinsey estimates that big data and the machine learning in pharma and medicine could generate a value of up to $100 billion annually. So this is a sector which will keep evolving. Okay. And the money spent on this sector is huge. Now, if we want to apply analytics in the pharma sector, we need a lot of data. Now, how do we get this data? Or where do we get this data? So the data generated from many streams in medical and related sector is very critical in developing innovations in pharma sector. Any analytics you want to apply, you need the past data or the data which was already collected and studied upon. This will help you build the various analytics or machine learning models from where you can derive insights. Okay. So in pharma sector, and machine learning is leveraging this data to derive new insights which get clear by analytics. Now, where all can we get this data? We can get this data from research and development. Now, a company might have studied thousands of molecules before producing paracetamol, or before selecting a drug called paracetamol. There might be thousands of molecules which might have been studied. And this data went waste because only one drug had come into the market. So the data from all these 999 drugs, which also exists in the pharmaceutical company, can be taken and analyzed for further innovations. There might be some other molecules which can be used for some other targets and cure some other diseases. So, using this data from other 999 molecules will also be very helpful, and this will shorten the time. Now, the time which was spent, like 10 to 12 years, which was spent on discovering new drug molecule, that time also can be reduced because you have already the 999 drug and the data. Just applying analytics to this, there's a lot of time and money being saved. Next, there are physicians and clinics. So the data which is being generated on the physicians and the clinics, the medical prescriptions written by the doctors, and even their tests, diagnostic tests. All this data also, if it is stored, and then you can apply analytics on this data and see, okay, if this patient's sugar levels are this much, BP levels are this much, and the patient's age, all that, if you see, you can understand how the disease progression is there. What are the drugs which were helpful in this condition? All this can be studied if we have the past data from the physicians and the clinics. Even the patient's data, wherein you're asking, patients are telling us about their side effects, about how they feel about the disease after taking the medicines, all this patient data, the hospitals and caregivers data, the records, the medical records which are on the hospitals, all this data can be collected and applied. Then data from the pharma industries. Now, when manufacturing drugs, when will manufacturing happen? Manufacturing happens after the discovery of the drug. After the drug is discovered, it goes into manufacturing, wherein the tablet, say for example, paracetamol, is being manufactured. Now, this drug will be studied for all the pharmacokinetic and dynamic properties, wherein they see how the drug is absorbed, metabolized, excreted, what is the bioavailability of the drug, all that. Now, this data from the manufacturing industry can be taken, and if analytics are applied on this, maybe you can search for new ways wherein you can increase the bioavailability. Increasing bioavailability is nothing but increasing the efficiency of drug. Or say, for example, if a paracetamol is taking 15 minutes to act after you take it, then maybe you can apply different methods wherein you're trying to reduce the ADME of this, or absorption, digestion, metabolism, and excretion of this particular drug, and maybe you can increase the time taken to create the effect as 10 minutes. So rapid absorption and rapid effect can also be done. So there are many different ways you can apply analytics in pharma. In the retail pharma stores, you can take the data from the stores. This data would be mainly invoice data or billing data, wherein these invoices tell you how many of paracetamols were sold, how many of diclofenac was sold, and all that. So this will help in stocking the drugs. So if you know there will be 100 paracetamols which will be sold, then and provide this data to the retail pharma stores, then they'll stock 100 paracetamols next, and this will increase their sales and without creating burden on them. So this is also a way where you can apply analytics to help retail stores. For all this, you need data from these various sectors. Now, analytics can be applied in various stages in pharma. Those would be descriptive, predictive, prescriptive. So we'll understand descriptive, predictive, prescriptive when we are actually dealing with the stages of analytics. But to just give you some examples, we are going to see what are these and how analytics is being applied over here. In descriptive analytics, we can apply epidemiology analysis. Okay. Here, the study is mainly on the epidemiology. It is nothing but the demographics. Now, some diseases are prevalent in some places. Okay, in specified population and specified place, a particular disease is there. So you're studying that and helping the pharma companies to produce more drug in that particular area. So that is about epidemiology. You're understanding if that particular country has a particular disease and producing drug specific for that. Then there is unmet medical need analysis. Okay, some countries there is a need for drug, but the drug was never innovated or discovered. So you can apply analysis so that you understand there is the requirement for the specific drug, and the study or the discovery process by the pharma company can be focused more on this particular need. So wherever there is need, they try to apply more analytics and discover. Then market opportunity assessment. In here, we apply analytics to see where is the opportunity. Now, I'm company A, pharma company A, and there are other companies called pharma company B and C. Now, as a pharma company A, I'm trying to see for an opportunity where I can produce particular drugs. So I see where all pharma B company has their units, manufacturing units, or which all market they are producing. And if there is any scope or an opportunity, then the other countries might be, as a pharma company, I might be producing the drugs. So understanding the market and the opportunity can also be done using the analytics. Now, moving ahead to predictive analytics. In predictive analytics, we are mainly developing forecasting models. So once there is a past data, we take that data and try to predict the future outcome. Okay. Patient-based forecast models are nothing but wherein the past data of the patient is studied, okay, and the demand and the success for those drugs are studied, and based on this, they predict the future requirement. This is based on patient, whereas then there is market-based forecast models, wherein they study the future market trends, and based on that, appropriate amount of medicines are stocked. Okay. So this is mainly in the forecasting area where you're predicting the future trends, future needs, and then uh, stocking it appropriately. Evaluating business development and licensing opportunities. Okay. So here you're evaluating how much the business can be developed. Like say in the next 10 years, where would be the business? How would be the growth? And where all you need to apply for license. Say you're manufacturing the drug in country A. Now you want to expand your business and manufacture that in different countries. So what are the licensing opportunities? Where all can be applied? All this can also be done using predictive analytics. Then there is portfolio analysis. So here, if there is a pharma company A, now this company has different medicines which are being produced. Now, in these medicines, they want to analyze which are the most ones, most used, and the most sold medicines. And so they are analyzing their portfolio, and are uh, the best ones are then decided, and this will help in best ones are decided, and this will help in allocating all the resources to this best drug and increasing the sales of this and increasing the quantity of this basically. Then the pricing analysis. Now, once a drug is discovered and is out to be put into the market, then what the pricing should be, how much would benefit the consumers also, and also the pharma company. So the pricing analysis based on a lot of things, like even the competitor rates have to be considered over here, even the ROI has to be considered. All these will be considered, and an appropriate pricing will be applied. So that is about pricing analysis. Then health economics and outcome research. Once the medical interventions are done, now there were um, medicines given or the medical devices used. What were the outcomes, and all that will be studied, and based on that, the cost effectiveness of that particular drug and its impact will be studied. So once this is analyzed, you can price the drug better and also increase the outcome. So these are the various applications of analytics in pharma, and these are just a few examples, but there are a lot. There's a lot of scope in pharma industries, like wherein you can apply almost analytics in every stage, and this not just affects in terms of money. Like if you apply analytics in pharma, firstly, there is improved patient care, improved, a return on investments, then improved efficiency, even the time consumed. Okay, you can reduce the time taken for all this extensive process that goes on in the drug discovery and development process. So all in all, in pharma and analytics together play a major role, and applying analytics to pharma requires some of the domain knowledge, which is why this particular sector will keep growing exponentially once you have the knowledge of both the domains. Now, what all are we going to cover in this course? After the introduction into the pharmacy, we are going to cover the data mining primer. And then we will be moving ahead with the machine learning, mainly the unsupervised learning methods, that is clustering. Then we will move ahead with the supervised learning, machine learning methods, that is regression models. We'll see the examples for each as we move ahead. Now, when we say analytics, we are trying to understand a pattern. Okay, we are trying to analyze the pattern in the data. Now, this data might be past data. Okay, this is a in general explanation of analytics. But when this analytics, like understanding the pattern, is mainly done on pharma data, then it can be called as pharma analytics. Okay, so say for example, you have a patient data. A patient is suffering from diabetes, and he has hypertension, he has BP, and then his age is 60, and he is on tablets like metformin and Olbisat. Now, in such a way, if you have the patient data of around 30 patients who are suffering from diabetes and having the age 60, and they are on different medications. Now, analyzing the pattern in this data, like at say for example, in this you can see because the age of the patient is high, he has multiple conditions like diabetes and hypertension. So based seeing other records also, you may come into analysis that with age, there is increase in the prevalence of the disease. Okay, so such patterns can be analyzed once you see the past data. Okay. Once we see this as the past data, we can tell, or we can come to conclusion that, okay, this particular drug might be helpful in diabetes, or this particular disease might be prevalent in a specific age group. So all these patterns can be studied from the past data. To study these patterns, to study all the past data, we follow a specific procedure. Okay, a step-by-step procedure which is followed so that we can analyze the data. We will be seeing that process also. Coming to stages of analytics. First, we will be covering the stages of analytics. Then we will be moving forward to CRISP MLQ. CRISP MLQ is nothing but the cross-industry standard procedure for machine learning, and then supervised and unsupervised learning and machine learning primer. Then we will be studying about the splitting of data, error functions, overfitting and underfitting. Okay. As I said, analytics is nothing but understanding the pattern in data. Okay. This involves multiple stages. Those stages are descriptive analytics, diagnostic analytics, predictive analytics, and prescriptive analytics. Okay. Now, in descriptive analytics, you are describing what happened. Okay. Mainly, you're answering the question, what happened? Whereas in diagnostic analytics, we are trying to understand why exactly something happened. And then in predictive analytics, we'll be moving ahead and trying to see what is likely to happen in the future. And in prescriptive, we will try to give a solution as to what is to be done. Okay. Now, coming to the first stage, that is descriptive analytics. Okay. Now, in descriptive analytics, we are trying to understand what happened. Okay. Try to remember these keywords, that is what happened in the past. Here, mainly the companies try to understand the trends and patterns within the past data. Okay. So in the data, whatever the pattern was, they try to understand that. If I have to give an example, say for example, a person falls down. Okay. Out of giddiness, a person falls down in front of you. And then you take that person to the doctor. Okay. Now, as soon as you go to the doctor, what will the doctor say? The doctor will ask you, what happened? Right? Then you will be telling him that the person fell down out of giddiness. Okay. This is nothing but descriptive analytics, where you are trying to understand what happened. The next stage would be diagnostic analytics. Okay. In diagnostic analytics, we are trying to understand why something happened. Okay. Once you go to the doctor and tell that a person fell down out of giddiness, then the doctor tries to diagnose why he fell down. Okay, why he had giddiness. It might be because he hasn't eaten well, or he hasn't slept properly, all these. So doctor is trying to diagnose what exactly, like why something happened. Okay, that is diagnostic analytics. So this goes beyond this historical data. Like here, you're trying to analyze why the past event happened. Okay, this will involve more data analytic techniques, like you're trying to understand or drill down what exactly happened, correlate it. Maybe because he hasn't slept or he hasn't eaten, he might have fell out of giddiness. So trying to diagnose why something happened. Once what happened and why something happened is clear, then we can apply, or the next would be the predictive analytics stage. Okay, what is likely to happen? Okay, in predictive analytics, the doctor would be telling you what would happen to that to the patient in future. Okay, like if the patient doesn't eat well, he is going to die. So like that, what would happen in the future or what is likely to happen is what you do in predictive analytics. Okay, so here you apply various statistics and forecasting techniques to understand what would happen in the future. Okay, future trends is what you're trying to see. Then there is prescriptive analytics. Okay. At the end, the doctor will prescribe you something. Okay, so that the person doesn't fall out of giddiness. Maybe he'll give some tonics for the energy, something. So prescribing. So, or what is to be done. If he takes the medicine, then he'll be all right. So the prescriptive analytics mainly answers or is mainly the decision making step wherein what has to be done is being analyzed. Okay. So overall, first is descriptive, which will tell us what happened. Then there is diagnostic, wherein we are trying to tell why something happened. Then there is predictive analytics, where we are trying to understand what is to be done. Sorry. What is likely to happen. And there is prescriptive, wherein we describe what is to be done. So these are the different stages of analytics. And all the stages can be applied based on the use cases. So particular stage, particular problem we are trying to address. Okay. So in describing, we are telling the doctor what exactly happened in the past. When diagnosing, the doctor is trying to diagnose why the person fell out of giddiness. And he's trying to correlate whether it might be due to not eating well or not sleeping well. Then there is predictive, wherein the doctor is trying to tell the patient if he what would happen in the future, and how the disease would progress. Then there is prescriptive, wherein doctor is trying to address the issue, like because the person is um, falling out of giddiness because of low energy, maybe the doctor might prescribe some of the um, energy boosting syrups. So these are the different stages of analytics. Basically, we are trying to understand the pattern in the data. For anything, the past data is very, very important. Now, there is a standard process called CRISP MLQ. This is the cross industry standard process for machine learning with quality assurance. Okay. There's a particular process which is followed so that the process of building these machine learning models gets easier. Okay. So to apply all the steps in process, we try to follow CRISP MLQ methodology. It's nothing but cross-industry standard process for machine learning with quality assurance. It involves 6 steps. Okay. First is business and data understanding. After understanding the business and data, we move forward to data preparation. Then we move forward to model building and tuning. Then once the model is built and tuned, then we go for evaluation techniques. And once the model is evaluated, this model will be deployed onto the software, and from there it would be monitored and maintained. Now, what exactly happens in these 6 phases and how the methodology is followed is what we are going to see next. Now, okay. So this is the mind map. I'll be explaining you the CRISP-MLQ methodology from here on. Now, CRISP-MLQ is nothing but the project management methodology. This is cross-industry standard process for machine learning with quality assurance. It involves the 6 steps. The first step in this methodology would be business understanding. Now, what is business understanding? Okay. Here, when you're trying to start this project, here you try to define what exactly the business wants, the problem the business is facing, and what do you want to solve. Okay. So, business understanding. Here, we record the objective and the constraint. Like, what is the problem we are trying to solve, or what would our objective would be? Okay. Then we move forward to business constraint. Like, in order to achieve this objective, what would be the constraint? The business objective and the constraint would always be written in the business optimization terms. Okay, that is maximize and minimize. Using these two terms, we try to describe what would the objective be, what would be the constraint. Okay. Then we also try to determine the success criteria. The business success criteria, ML success criteria, and economic success criteria. In business success criteria, we try to tell how we are going to help the business. This is always in measurable terms. The success criteria will always be written in measurable terms so that we can describe how we are going to achieve the success. Say we want to increase the sales of a particular drug by 10%, 20%. And in ML success criteria, we are trying to define how much accuracy or our machine learning model would be how much accurate. Okay. So in machine learning success criteria, we are trying to define how much accuracy we are expecting from the model to and how much accuracy we are going to achieve. In economic success criteria, I'm trying to tell in monetary terms how we are going to be helpful for the business. Okay. So now we write the objectives and constraint using only 2 to 3 words, and that too we use the optimization terms, that is minimize and maximize. These are objectives, constraints, and all the business understanding things will be written in the project charter. So, project charter would be the first document which gets prepared on any project, and it is containing high-level details. Okay, the project characteristics, requirements, milestones, what is the budget allocated, who are the key stakeholders, and the risks. So all this will be written on the project charter, and this document will be signed by the project sponsor. Once this particular document is signed, then the actual project starts. Okay. So basically, in business understanding, this would be the initial phase, which focuses on understanding the objectives of the project. Okay, the objectives, requirements, and this would all be in the terms of business perspective. How it would help the business. Then converting this knowledge into data mining problem definition, and a preliminary plan is designed to achieve these objectives. So this would be the plan. This is all what would be written in the business understanding stage. Okay. Once you know that this is the problem a business is facing, and you know that this would be the objective you would try to achieve, then you will be moving ahead to collect and understand the data. So the next would be data understanding phase. Okay. Now, say if we take pharma example only, say the pharmaceutical company is trying to increase the sales of a diabetic drug called metformin. Now, they want to increase the sales. This is the objective, or you're trying to maximize sales of this drug, and the constraint would be not increasing or not spending too much. You want to maximize the sales. For this, to achieve this objective, first you will have to collect the data and understand. So you will be moving ahead to data collection phase. If we are trying to study a diabetic drug, then the data which we collect should also be related to that, right? So from where can we collect this data? Data collection is again uh, multifaceted. Like you can collect it from different sources. One is primary, another is secondary. Okay. So basically, data understanding phase starts with an initial data collection and proceeds with the activities to get familiar with the data, like to identify data quality problems, to even understand the first insights into the data. Okay. Or to detect interesting subsets to form hypothesis for hidden information. Okay. Now, if we want to understand the sales of metformin, which is a diabetic drug, then we can collect this data from different sources. One way would be to collect at the source, or like to collect from the pharmaceutical company, which we are solving the problem for. Okay, data can be collected at source or from the secondary data sources. Primary data sources are nothing but data collected at source. This could be data collected from the surveys, from experiments, or from simulation. Okay, you simulate the same effect and try to collect the data, or you can scrape the web, extract from social media, or you can take it from the IoT sensors if it is in case of manufacturing industries. Okay. Now, the advantages of collecting the data at source would be that you can get the data for exact variable of interest you want. So if you want the data for metformin, then you can get the data exactly for metformin tablet. But it is costly and time consuming. Then there are secondary data sources. These are data which were collected beforehand. Okay. This data was already collected or it was already stored for some other purposes. This could be the data which is stored in the relational database management systems. So the data which was already stored in the databases by the pharma companies or any person who has stored it, and it is publicly available, open source as well as syndicate data. So this would be secondary data source. Okay. In both ways, whichever way is feasible, we can go for collecting the data. And once we get the data or variable of interest, then we can proceed with the further steps. The plus point for collecting data from secondary sources is that it is easily available. As it is open source, it is easily available on the websites also. So you can collect it easily. But the problem is you may or may not exactly have the variable of interest. Like if you want data related to metformin, and if you go search it online as an open source, you may or may not have the metformin data exactly, or say with the metformin, you want age data. So this particular column may or may not be there, as this data was collected for some other purposes, but you are using that data for your analysis. So this would be secondary data source. Next, there are different steps again. Maintaining the data version, the describing the data, like when you collect the data, what data holds, like that units, the format, all that you're describing over here. Then the requirements, as in how you do, then verification of the data. So all these are the different steps in data collection. Okay. This particular methodology, that is CRISP-MQ, which has 6 phases, this can be applied to any process. Okay, this is a product management technology. This product management methodology, and this can be applied to all the data analytic projects. How we can apply this for our pharma data is what we are studying here. Now, once you have collected the data, you'll further analyze, or the type of the data is also important. Okay. Now, there are different types of data. We'll be studying a few of them. There is discrete data, then there is continuous data. Okay. Continuous data would be data which can be represented in decimal formats also, like height, weight, all this numerical data, which when you represent it in decimal format also, it makes sense. That would be continuous data. But discrete data is something, if you try to represent in decimal format, it does not make sense. Okay. Discrete data would be mainly categorical data. Okay. There might be categorical. The examples would be red, green, and yellow. Okay. If the data has particular categories. Now, red, green, yellow is a particular category, or gender, which is male and female, are 2 categories. So this way, if the data has different categories or categorical data, then it comes under discrete. Now, there is also count data. Count data. Like if I say how many laptops are there. So this particular count data can also come in discrete data. Say if I say there are 7 laptops, it makes sense. But if I try to represent this in decimal format, say 7.3 laptops, this doesn't make sense as you can't divide that laptop, right? So only the count data can be in discrete. The rest all would be in the continuous data. So there's 2 types, discrete and continuous. Then there is qualitative versus quantitative data. Qualitative describes the quality and quantity describes the measurement. Then again, there is a structured, semi-structured, and unstructured data. Structured data is something which you can represent in the tabular format. Okay, structured data is always in the tables. This data you can call it as structured data. Semi-structured data is something which is not in the tabular format, but if you try to put it in the table, you can easily transform it. Okay, you can take this particular data and put it into the table. This is semi-structured data. Moving ahead with unstructured data. So this unstructured data cannot be placed in raw state in the table. Okay, you cannot place the unstructured data into the tabular format. This can be videos, images, audio, and textual data. Moving ahead, there is a big data and non-big data. Big data is something which has the four ways. That is volume, velocity, variety, and veracity. Volume would be the data which has huge volume. Okay. Velocity is something where data is being generated at a rapid speed. Then there is variety, wherein there is videos, images, audios, text, all kinds of data is there. Then there is veracity, where there is uncertainty in the data. Okay. So the data which has these V's, okay, any of these or all of these, then that can be called as big data. Then there is cross-sectional, time series, and longitudinal data. Okay. When it comes to cross-sectional data, here you are trying to take the data at a particular instant of time. Okay. So you have multiple columns, but here you are not considering the date and time. Whereas in time series data, time is very important. Okay. So with respect to time, you're collecting the other data. Okay. With the date and time, you're collecting the all other columns. Then there is longitudinal data or panel data, which is both cross-sectional and time series. So it has variable columns and also the time column. This is nothing but the longitudinal or panel data. It combines both the cross-sectional and time series data. Balanced versus imbalanced data or rare events. Okay. Whenever we are taking the data and pushing it into the model, if there are multiple classes, you always try to balance the data sets. Okay. If there are two classes, then both classes should have equal number. So if there are balanced data sets, we can go for simple random sampling. If there are imbalanced data sets, then we apply different methods to balance the classes. Okay. Then there is offline data and live streaming data. Okay. So what all did we do here? First, we try to understand what the problem was. Okay. What was the business problem? We defined the objectives and the constraints which we are trying to achieve. Once this was done, we moved ahead to collect the data, and also we studied different data types. In what all types does the data exist? Okay, according to our use case, we can collect the particular data type. They can be continuous, discrete, big data, non-big data, all that. Qualitative, quantitative, etc. Okay. Now, this was about data understanding. Both these come under the first phase of CRISPR-MQ. Okay, 1A and 1B. Once you have the data with you, next you will try to prepare the data. Okay, this data preparation again has different steps. Okay, there is exploratory data analysis or descriptive statistics. Then there is data cleansing, data preparation, data munging, data wrangling, or data organizing. Then there is feature engineering. Okay. Now, Okay. Coming to the data. Now say you have collected the patient related data in different columns. Okay. Now if you have to analyze this data, it becomes easier once you have the data in a particular format, right? So we try to clean or prepare the data so that we don't have any disturbances in the data. Okay. This particular step can be called as data cleansing, data preparation, data munging, data wrangling, or data organizing. Okay. Now let us first see how to cleanse the data and then move ahead with the EDA. Now, there are around 10 steps which are involved in cleaning the data. You need not apply all the 10 steps. Based on the data which you have, you can select the step which is appropriate and work on it. Now, say you have collected the data and there are some disturbances in it. Now, you're trying to apply these particular steps to clean it. One would be typecasting. Here, you're trying to change the data type of the data. Okay. So if the data sometimes is in categories, okay, say yes, but sorry. Let me take some other example. Now, if the data is an integer, like 1 or 2, and you want to change this data type into decimals or floats, then you apply typecasting and change it into decimal or float. Okay. Then there are duplicates in the data. Sometimes the data which you collect from the pharma companies or the patient data which you collect might have duplicates. The same record might be repeated in different data sets you have collected. Such cases, we try to handle the duplicates. Okay, only one original record you want to keep. You want to get the single source of truth from various entries in one such example. Okay, for this you can go for data.duplicated. Okay, so if there are duplicates in the data, we can handle them. Then there are outlier analysis or treatment. So here, outliers are nothing but extreme points. Okay, sometimes there might be outliers in the data. Say for example, you have age column. All the patient data which you have has a age from 2 years to 80 years or something. But there is one record, 1 or 2 records, which have age like around 200, 300. This is like not possible, or sometimes there is a data which has 100, 150, like that. So these are some extreme values which may or may not be possible, but this will affect our data or affect our analysis. So we are trying to analyze the outliers and either treat them or trim them. Okay, we are either removing those outliers or treating them. That is nothing but the outlier analysis or extreme value analysis. Next, there is zero and near zero variance features. Here, some columns might have only one unique, only one value. Say, for example, the country column in particular data has only USA in the whole data. So there is no variability over here. All the records are same. When all the records are same, there is no use keeping this particular column. So basically, the columns which have zero variance, okay, but there is no variability in the data, then we try to remove such columns or ignore such columns. So that is zero variance columns. Then there is discretization, binning, or grouping. Sometimes the data which is continuous, you want it in discrete, means the data which is in numbers, you want it into the categories. Okay. Okay. Like this, if you want to keep them in the categories, then you can go for discretization, binning, or grouping. Either you can fix it with particular bins, like particular intervals, or convert it into binary categories, or you can even round them. So all these different steps are to discretize, bin, or group the data. Then there is dummy variable creation. Okay. Here you're trying to convert discrete data into continuous data. You have categories, you're converting them into numerical form. There is one hot encoding, then there is label encoding. And there are different methods also. Then the data might also have missing values. If you have collected a patient data, okay, age, country, name. So some records might have all the columns, all the values, but some records might have missing values. Okay, so in such cases, we need to treat the missing values. So missing value treatment is also a way of preparing the data. Here we are trying to impute these missing values. Okay. Impute means we are putting those values over there. How do we input these values over here? We can even go for mean, or there are many other techniques like mean imputation, median imputation, mode imputation, random imputation, hot deck imputation, regression imputation, or KNN imputation. So when there are missing values in the data, we can go for different techniques. One would be to take the mean. Say you have a 30, 40, 50, and there is one missing value. So taking the mean of these three numbers and imputing the value for the missing value would be mean imputation. Then there is transformation, wherein you are trying to check if the data has normal distribution. Not you're applying transformation techniques. Then there is feature scaling or feature shrinking, wherein you try to make the data scale free and unitless. Okay. Why do we want to make the data scale free? Say, for example, you have patient data, and this data has multiple columns. One is age, one is height, there is weight, gender, all that. Now, age is in different format, height is in different, weight is in different. If we try to compare all these columns, because they are of different units, we will not be able to compare. So we want to make this data scale free and even unit free. Okay, for that we apply feature scaling or feature shrinking technique. It can be standardized scaling, min-max scaling, or normalization, or robust scaling. Okay. Standardized scaling would be nothing but making it into standard format. So this will be in the range of -3 to +3. Whereas a normalization would be making it into 0 to 1 range. Okay. Then there is robust scaling, which is mainly applied when there are outliers or extreme values in the data. X minus median by IQR. So this is feature scaling or feature shrinking. Then there are string manipulations. This is mainly for unstructured textual data. As I said, structured data would be something which is in tabular form. And then unstructured data is something which cannot be put into tabular form. Now, textual data, which is unstructured, for this data, you can apply the string manipulation techniques. That is tokenization, stemming or lemmatization, stop word removal, document similarity, topic models, and word embeddings. More about this we will be seeing in the tomorrow's session. Now, once you have applied the steps, okay, once you have prepared the data after applying the appropriate steps from these, you're going for exploratory data analysis or descriptive statistics. Now, there is different business moment decisions. That is first moment business decision, second moment business decision, third moment, and fourth moment business decision. Here, you're trying to understand the pattern in the data. You're exploring the data. Okay, first moment business decision will hold all the measures of central tendency. That is mean, median, mode. So it will check all the measures of central tendency. Mean is nothing but the average. Median would be the centermost or middlemost value, and mode would be the value which repeats maximum number of times. So why do we see these first moment business decisions? This is nothing but helping us understand the data at the first glance. As soon as you see the data at the first moment, you try to understand these particular uh, metrics. Okay, all these things you can just understand it in the first glance. Next, we will move forward to second moment business decision. First moment was just calculating the average or the middlemost value or telling us the value which was repeated multiple times. Then there is second moment business decision wherein we are calculating the measures of dispersion. Okay, dispersion in the sense how the data is dispersed. Okay, how it is spread. This is mainly calculated from the second moment business decisions. This involves variance, standard deviation, and range. Okay. Now, the disadvantage of variance is that the units gets squared. So that is why we try to apply standard deviation, wherein we are taking the square root of variance and trying to understand. Here we don't have this a disadvantage of units getting squared. Okay. Next, there is range. Range is nothing but the maximum value and the minimum value. So overall, by understanding the measures of dispersion, we are trying to see how the data is distributed. What is the maximum value, minimum value? What is the spread of it? How far is it from the mean? If this is mean, how far is the data point from the mean? This is nothing but the variance. And if you try to take the square root of the variance, it is standard deviation. Okay, these are nothing but the measures of dispersion. Moving ahead, there is third moment business decision, that is skewness. Skewness gives us the asymmetry in the data. Okay, asymmetry in the distribution. Okay, skewness is given by these tails in the data. So this will show us the asymmetry. There is right skewed, left skewed, and normally distributed data. So skewness is telling us how if the data is distributed normally or if there is any skewness or asymmetry in the data. Then there is fourth moment business decision, which is kurtosis. Kurtosis tells us the peakness in the data. Okay, if the data is distributed like this, how peaked it is, where the peak is. Okay, so There are three types, that is, if it is mesokurtic or normal distribution, then the kurtosis would be 3. Then there is platykurtic or negative kurtosis. Then there is positive kurtosis or leptokurtic distribution. Okay, there's a sharp peak and thick tails. So kurtosis will show us the peakedness in the data. Then we can represent this data graphically also. There are univariate, bivariate, and multivariate analysis. Univariate, where you're plotting only one variable on the graph. So that would be univariate analysis. Bivariate would be taking two variables into consideration and plotting. This would involve scatter plot. Then there is multivariate, where there are multiple variables being plotted on one plot. That is nothing but pair plot and interaction plot. So by doing all this, the first moment business decision, second moment, third moment, and fourth moment, we are trying to basically get a picture of how the data is, like its distribution, how the data is spread, where is the data more concentrated upon, to get an overview or how to understand how the pattern is. We try to go for exploratory data analysis. Okay. Now, these few steps can be applied directly, or you can go for auto ADA. Auto ADA are again libraries in Python which will help you to apply all these steps in just one line of code. So the different auto ADA libraries would be the Sweetbus library, AutoVis library, Detail, Pandas profiling, data prep. And there are many more libraries which are getting added upon. Okay. So in auto EDA libraries, you can just take the library, write one line of code, and all these business momentizations would be calculated in one go. Okay. Next, there is feature engineering. Okay. Feature engineering can be done on multiple data. Okay, temporal data or numeric data, categorical data, text data, image data. So how you want to select and extract the data, all this can be done in feature engineering. So this is whole data preparation phase. Okay, here we are trying to understand the pattern and also clean the data. Once all this is done, then we can move ahead with the model building part. Okay. What is model building? Building these algorithms or data science models, this can also be called as data mining or machine learning. You might have heard the word machine learning, right? Machine learning is all these models. Okay, so in this phase, we are going to see what are the different machine learning models and even the primer. Okay, how to build these, all that. So this is the core step. That is, once the data is prepared, then you build the model according to the data and then go for analysis. Okay, in depth about this, we will be studying in the next phase. Let me move ahead with the evaluation. Once you have built a model, now you have to evaluate or check if the model you have built is appropriate or no. You want to check the accuracy of the model. So there are different accuracy measures which can be calculated based on what kind of problem you are solving. Okay? If it is a classification problem, if it is a regression problem, based on that, there are different accuracy measures And also there are different error functions. Okay. If y is continuous, these are the different accuracy measures. Then y is discrete. Then there are different accuracy measures. We'll be seeing these one by one. So there is accuracy and there are also error functions that can be calculated to evaluate whether the model we have built is appropriate or not. Then there is model deployment. Okay, whatever model you have built, you are now integrating it with the software. Okay, so that is nothing but model deployment. Once you are deploying the model, you will be monitoring it and maintaining it. Because as the new data comes up, the model's accuracy changes as it is trained on very less data. So you have to keep retraining the model. So this phase comes under monitoring and maintenance phase. Okay. Let me move back to the PPT. As we saw, CRISPR-MILQ has 6 phases. First phase has business understanding and data understanding, where we are trying to understand what is the business problem, objectives and constraints, and then we move ahead for data collection and understanding the different data types, which has to be collected. Next, there is data preparation phase. Once you have data in your hand, then you process or prepare the data. Okay, preparing the data by cleaning it and even applying different exploratory data analytics. Okay, you're trying to find the pattern in the data. This EDA can be done by various steps, that is first moment, second moment, third moment, and fourth moment business decisions. And you can even apply feature engineering methods. So overall, by these different steps, you are trying to prepare the data. So the data will be ready to put into the model building phase and tuning. Okay. Once the model building is done, we can evaluate if the model is appropriate or no. Based on the evaluation, choosing the best model, and this best model will go into the deployment. Okay. Once the model is deployed, then there is a need for regular monitoring and maintenance as the model accuracy may decrease due to time, and then you will have to retrain the model for the new data which it will be getting once it is with the client. Now, when it comes in model building phase, there are two types of machine learning algorithms. Here, mainly you're building algorithms. What kind of algorithm should you apply? There are so many different algorithms. So, what kind of algorithm would you apply? Again, depends on the data you're using firstly. Second, the use case you're solving. Okay. So, and the machine learning algorithms, there is unsupervised learning and there is supervised learning. Okay. Now, when it is unsupervised learning, it involves training a model on data which doesn't have labels. Okay. The data which doesn't have labels in it. Those data you can go for unsupervised learning. Labels in the sense the output variable. Okay. There are no labels, then you can also apply unsupervised learning. Here, the model will try to understand the patterns. Okay, it is taking the data you're giving it and it is trying to understand the patterns on its own without any guidance. Okay, and then it will group the data. So, overall, unsupervised learning involves training model on data that has no labels in it. Okay, there are no labels in the data, and the model tries to identify the patterns without any guidance on how the outputs should be. Okay, so if there are no output labels, then what is the goal of the model or the unsupervised learning? The goal is to explore the data and find some structure. Okay, so here we are just trying to find some structure or pattern within the data. So these algorithms try to model the underlying distribution of the data or find the relationships between the data. Once you find the relations, you can group them. Okay. Then in unsupervised learning, there might be clustering techniques, dimensional reduction, associations, etc. Okay. Like in clustering is a type of unsupervised learning. In clustering, we are taking the data and we are grouping them. Okay, we're just grouping them into different clusters. Next, in dimension reduction, we are trying to reduce the dimensions of the data consideration. Then, in association rules, we are trying to discover the relationship between the data points. Okay. Here, so unsupervised learning can be used so that we can discover some hidden patterns in the data. And you don't need any labels when you're applying the unsupervised learning. This is only applied when you want to learn the pattern. That's all, not predict. Okay. So the flow of unsupervised learning would be training the taking the historical data. Okay, past data is being taken. This data will be sent to the uh, model or machine learning algorithm. This model will apply and get the rules or patterns. Okay. So all these things it will do on its own. Next, when you do the prediction, when you give the new data, it will try to apply these rules and patterns and see which category that particular uh, data point belongs to. So basically, it is trying to understand the patterns on its own and then see on the new data which pattern or which cluster it belongs to. Okay. So unsupervised learning models work without any supervision and learn on their own. Next, there is supervised learning. Okay. Supervised learning is applied when you have output variable in the data. Okay. So first you are training the model on the data set. Okay. This data set will have the output variable too. Now, once you train the model on the data set which has the output variable also, then the model will be able to learn the patterns. Okay. Now it will try to create a equation or learn the pattern, wherein next when you try to give it a new data, then it will be able to predict on that new data also. Okay. So in supervised learning, the model is trained on a labeled data set. And that meaning that each training example is paired with an output variable. The model learns to predict the output from the input during the training process. Whatever input you're giving, it tries to learn. The goal of supervised learning is to predict outcome for new unseen data. Okay, when a new data is passed, then you can predict the outcome. So it is basically trying to understand the relation between the input variables and the output variables. Now, say if you have multiple columns in your data set. These are your different columns, and this is your output variable, whatever you're trying to detect. Okay, basically, you are giving this whole data to the supervised learning model and trying to train it. So, the model will be trained. That is for this output variable y. It will see the relationship between x4, x3, x2, and x1. Will come up with an equation. Okay, by finding the relationship between the input variables and the output variable. By finding this particular relationship, it will be able to predict outcomes for the new data. Now, when you give new data, okay, when the client gives you some data which is new but has the same columns x1, x2, x3, though you don't have y output for this, this particular model will be able to predict our output or the outcome. That is supervised learning. Okay. In supervised learning, we are giving the historical data or past data, which has the output variable y also. Okay. Then you sample it. Sampling can be random. Okay. Random sampling, or there are some other methods to sample also. So once you sample it, you are sending some part of the data to training and some to validation. Okay. Say you're sending around 80% of the data to training, 20% of the data to validation. Once you train the model on 80% of your data, it will learn the equation, the mathematical equation or the relationship. We'll find out, and then when you send the validation data, the 20% of the data into validation, will try to predict using this equation. Whatever the predictions are, that is compared with the actual values and the model is validated. Based on this validation outcome, that is the accuracy, the model is seen if it is performing well or no. And if it is not performing well, then the hyperparameters are adjusted. This increases the accuracy. Once you know that the accuracy is improved and that is the best model, then you're taking the model and giving it new data. Then the model will take the equation and predict the outcomes. This is supervised learning. Okay, taking the data, sampling it, sending around 80% of data into training, 20% into validation, then training the model. So here you're sending a y variable also, output variable also. So the model will be able to get an equation. Based on that equation, it will be able to predict the future outcomes. Now, this equation again is validated on the validation data set. That is 20% of the original data set. This will be validated, and based on the accuracies, the hyperparameters would be adjusted and the best model is determined. Once the best model is determined, you test it on the new data. Okay. Overall, the goal is to predict the outcomes for the new or the unseen data based on learning from the training data set. Okay. Basically, it is trying to map inputs to the outputs. Now, what kind of problems can be solved? There might be classification problems, there might be regression problems. Like in classification problems, we can take the example of emails, classifying them as spam or not spam. Okay. So our y variable or the output variable would be spam or not spam. So here you will be giving a lot of data, email data with the output variable. 80% of this data will go into training, and you will train the model telling that if the mail has these particular words, then it is spam. If not, then it is not spam. So once the training is done, now if you give this model new data, a new email, you show it, and it will be able to predict whether that email is spam or not spam. This is a particular classification type of problem. Then there are regression problems wherein the output value is continuous. In the previous example, our output value was spam or not spam. If it is continuous, like numerical data, like predicting temperatures or predicting prices, then you can apply regression techniques. Okay, like you are trying to forecast the house prices based on their characteristics, then you can apply this supervised learning techniques. The advantages of supervised learning is it allows for direct forecasting of outcomes based on historical data. And it is easier also to measure the model's performance. But the problem is that every time you want to train a model, you need labeled data. And also, the supervised learning models have a quality to overfit. Okay, they are prone to overfitting. So if the models or if the data is too complex, then the supervised learning models are prone to overfitting. So how do we choose whether we want to go for supervised learning or unsupervised learning? So that depends on the business problem you are trying to solve. Okay, whether you want to, predict the future or just cluster or group the data based on the business problem you are trying to solve, you can either apply unsupervised learning algorithms or supervised learning algorithms. Okay. Now, splitting the data in supervised learning. Okay. How do we split the data and then how do we tune it? Whatever the data is, say the data is 100%. Okay. Now, some part of this data will go into training, some in validation, and some in testing. First, you train the model on, say, 60% of the data. Okay, if this is 100% of the data, you're sending 60 into training, 20 into validation, and 20 into testing. Okay, so first you're sending the data with the output variable, you're sending the data to train. Okay, once the training is done, you're building a model. Now, you check the accuracy. And then you also send the 20% of the data into validation. Okay, here you check the validation accuracy or error. If the accuracy is low or there is high error, then you are adjusting the parameters. There are many parameters in each algorithm. Those parameters, if you try to adjust according to the use case, then you may increase the accuracy. This particular step, wherein you are fine-tuning the model, is called as hyperparameter tuning. Okay. So until you get the better accuracy, you try to adjust the parameters. This is a validation. Okay. Fine-tuning the model parameters to get better accuracy. And then pick the best performing algorithm. Okay. So check adjusting the parameters, retraining the model. Adjusting the parameters, retraining the model. So you will do this until accuracy of both is better. Okay, when accuracy increases and error decreases, then you pick the particular model as the best performing algorithm. Okay. Once this is done and you have got the better accuracy, you go for testing. Here you are testing the data to get the test accuracy, also called as, and also the error or generalization error. Then you try to check if the model is overfitting or underfitting. What is the concept of overfitting and underfitting? Now, you have trained the model on a little, say, some amount of data. Now you have got some accuracy. Okay, 95%. Then, if you're testing the model, then also your accuracy should be somewhere near to this figure. Okay, if the train accuracy is more and the test is less, okay, meaning when you're trying to train the model on data, its accuracy was good, meaning it was performing well. But once you try to see the accuracy on the new data is nothing but the test data, the model doesn't perform well, the accuracy decreases. Okay, in such cases, the model is said to be overfitting. Overfitting is nothing but where the model has got trained well on the training data, but is not able to predict well on the test data. This particular condition is called as overfitting. Now, there's another case wherein model performs poorly on train data. That is, train accuracy is less. And test accuracy is also very less, because if the model is not trained well on the train data only, then there's no way it can predict well on the test data. So if the model has poor performance in train data, then it is called as underfitting model. Okay. Both the conditions, overfitting and underfitting, are not good. So the model should neither be overfitting nor be underfitting. We want a model which is right fit. Right fit model is the model which has training accuracy and test accuracy almost in the same range. Now, this was about unsupervised and supervised learning. Once you have trained the model, then you check the model for the accuracy and the errors. Okay, this is nothing but model evaluation. Okay, this is another phase of CRISP MLQ, where you are checking if the model you have built is good or not, appropriate or not. This is based on a lot of evaluation metrics. And these evaluation metrics also depend on the model you have built and the use case you are trying to solve. Okay. If, for example, your Y is categorical, okay, there are different categories. Spam, not spam. Like that. Or the diabetic, non-diabetic. Okay, these are the different categories. Now, how do you evaluate if the model you have built is working well or no when there is categorical data? For that, we go for confusion matrix. This particular matrix is called as confusion matrix. Okay. These would be the actual values and this would be the predicted values. Now you are trying to compare how well the model has predicted. And based on that, there are different metrics. Okay. Now, let's take this confusion matrix over here. So here, this are the actual values. These are the predicted values. Actual values means the actual value of the patient or the ground truth. Now, a patient who has the disease is present. Okay, and even the model, the algorithm you have built has the model you have built has also predicted that the patient has disease. This particular condition can be called as true positive. Okay, this is right prediction. When the patient doesn't have disease, the disease is absent, and even the model has given that it is absent, then that is true negative. Okay, this is true positive. Next is true negative. Okay. These are right predictions. There are conditions where the disease is absent in the patient. The patient doesn't have disease, but the model predicted that it is having the disease or test is positive. This can be called as false positive or type 1 error. Okay. Now, the disease is present in the patient, but the model predicted that this is absent. So this is false negative. Okay. These two are right predictions. These two are wrong predictions. Now, using this confusion matrix, we can calculate different metrics. Okay. Now, first is accuracy. How do we calculate accuracy? We take the correct predictions by all the predictions. So, true positive plus true negative by true positive plus false negative plus false positive plus true negative. So, all this together will give us the accuracy of the model. If you want to calculate the error, then take the false negative and false positive and divide it with all the values. That is false negative, false positive, true positive, true negative. So we have to keep in mind that accuracy should always be greater than percentages of majority class. These are accuracy and error. Then there are other error functions, that is precision, sensitivity, specificity, false positive, false negative, F1 score, all these. What is precision? Precision gives us a measure wherein we are understanding how well our model is able to predict. Okay. Predict a patient having a disease as having disease. It's like predicting the true positives. Okay. Probability of correctly identifying a random patient with disease having a disease. So it is giving us the rate of positivities. So true positive divided by true positive plus false positive. So TP divided by TP plus FP. This would be giving us precision. Okay. So precision is very important, especially in healthcare, because If a patient is having disease and you are predicting it properly, then it is good. But if a patient is having disease, but the model is predicting false negative, then it might harm the patient's life. So precision rate and is very essential when it comes to healthcare, as you don't want any patient who has a disease to not be treated for the disease. Okay, so that was precision. Then there is sensitivity, recall or hit rate, or true positive rate. So this rate will give us the people who had the disease and were correctly identified as having the disease. Okay, so these both are the people who had the disease, but only TP or true positives were the ones who were correctly identified as having the disease. So this will give us sensitivity or recall. Okay, people who were correctly identified as having disease. Then there is specificity. This gives us true negative rate. That is, people who didn't have disease which were correctly identified as not having the disease. So true negative by true negative plus false positive. Then there is FP rate, which is given by 1 minus specificity, false negative rate, which is given by 1 minus sensitivity. Then there is F1 score, which is calculated by 2 into precision into recall by precision plus recall. Okay, if the F1 score is 1 to 0 and defines a measure that balances precision and recall. This all is when your Y is categorical. Now, if the Y is continuous, meaning if the Y value is numerical, then we go for these measures or these error forms. That is mean error, mean absolute deviation, mean squared error, root mean squared error, mean percentage error, and mean absolute percentage error. Give me a minute. Okay. Now, when the Y is continuous, then the errors can be calculated in various ways. Those are mean error, mean absolute deviation, mean square, root mean squared, mean percentage, and mean absolute percentage error. Okay. This is mainly calculated by taking the actual value minus predicted value. If Y is your actual output variable and Y hat as your predicted value, Y minus Y hat will give you the error. Okay, so each error is again different. Mean error will give you Y minus Y hat whole square. Mean absolute deviation will take the absolute value Y minus Y hat. Okay, meaning only the positive value it takes. Okay, then there is mean squared error, Y minus Y hat whole square. This will square the units. Root mean squared error will take the square root. So the squared units problem is solved in root means squared error. Then there is mean percentage error. Here you're taking the percentage of the error. Next, Mean absolute percentage error, where you are calculating the error based on the predictions. Okay, minus y hat by y. Now, yeah, you might have a doubt like when to use which kind of error and what is this significance. So each error has its own significance, and choosing the error also is again problem specific. Okay. So basically mean squared error will take the square error, like it will square the error, and root mean squared error will take the square root of it. Okay. So this will avoid the bigger numbers, okay? Because you're taking the square, you are not giving weightage to the errors. So it is root mean square error is more interpretable in the same units as the target variable. It is actually sensitive to outliers also, and it might provide measures of how large errors are spread out. Okay, mean absolute error is more uh, robust. Like, okay, it is more robust to outliers when compared to mean squared error and root mean squared error. Then there is MAPE, that is mean absolute percentage error. Okay, here you're mainly taking it in the percentage uh, errors. Okay, it is measure of prediction accuracy, a forecasting method in percentage terms. This is useful when you want to interpret error as a percentage of actual values. Okay, like when you want to calculate or understand the error which is relative to the size of the actual value, then you can use mean absolute percentage error. This is mainly used in forecasting models. Okay. Now, moving ahead. As we had discussed, there is overfitting, which is nothing but variance, versus underfitting, which is nothing but bias. Okay. When you train the model and test it, there are chances where the model tends to overfit. To avoid this, you are trying to apply hyperparameter tuning. Okay, you see the test accuracy and valid accuracy, and then try to adjust the hyperparameters so that the model doesn't overfit. Okay, this can also be called as tuning the model or hyperparameter tuning. Okay, when we say model is overfitting, what does it mean? Now, if these are your data points, okay, if this is your data and you have predicted the regression line, or the predicted line would be this, means there are some values or some data points which are way far from the predicted data point. So this is not able to capture the pattern. Now, there is another model you have built wherein it has given this as the predicted line. Okay, these are your data points and this is your prediction line. The model seems to overfit the data points. Now, if this is fitting the training data point like this, when you pass a new data point, which is somewhere here, the model will not be able to predict well because it is overfitting on this data. Okay. So we want a model which is always right fit. Okay. These are the data points. If this is the predicted line, then this model will be able to predict well. So this is the right fit example. This is when the problem is a regression problem. When we have a classification problem, okay, now these are the data points and we are trying to classify these. Okay. If we classify this like this, then it is not able to classify it well. Some of the x's are on the line. Okay, this is one class and this is one class. Then, if these are my data points, and my predicted line would be like this, and it is fitting the data wherever I need. So if it is trying to fit it like this, overfit the training data, then if I give a new data point somewhere here, it will not be able to generalize or predict it well. So we want to avoid this overfitting. And we want a model which is a right fit. So it should be able to classify. Few errors would be fine, but this will be a more generalized model. So this is classification problem. Whenever we are trying to build a model, it should be either it should be always right fit. It should not be overfitted or underfitted. Overfit would not be generalized, whereas underfitting would not be able to predict the data. Okay. So basically, we split the data randomly so that we avoid bias. Okay. Whenever we take the data and put it into the model, we try to split it randomly. So as to not being biased to one particular class or particular data points. We can either take 60% into training, 20 into valid, and 20 into testing, or we can take 80% into training and 20% into testing. Okay. If there is large test set, then estimate future error as accurately as possible. Then if there is large training set, that is more better estimate. Okay. Now, by the end of this, we have learned about the differences between the supervised and the unsupervised learning. We have learned about the preliminaries and the machine learning models, and we have also learned how to partition the data, learned about the different errors, cost, or loss functions, and learned about overfitting and underfitting. Okay, this was about CRISPR method. Now, let us move to our first algorithm, that is unsupervised learning. And in unsupervised learning, we'll be moving ahead with k-means cluster. Okay. So the agenda would be to cover the machine learning in pharma industry, unsupervised learning that is clustering, and then k-means clustering. And we would be covering the advantages of k-means clustering, limitations of k-means clustering. Now, how can we use machine learning in pharma industry? By using machine learning in pharma industry, we can improve medical diagnosis. Like once we find the pattern between the input and the output variable, we can improve the diagnosis. Like we can try to diagnose the disease even before it occurs. Like basically, we can go for genetics, okay, using genes as a example. Like if a person has this particular gene, then he might be at the risk of developing a particular disease. So like that, we can help in building models which can help in improving diagnosis. Okay, we can work on betterment of patient care. Huh? If there are many drugs which are interacting with each other or which have a lot of side effects, then we can work on how to alleviate or reduce those effects and increase the patient care. Then we can even build models to predict when the next epidemic would happen. Okay. And we can build models to combat cancer. Then we can go for drug efficiency detection. Now, as I said, we already if a pharma industry was into a pharma company was into research and development, and they had studied around thousands of molecules. Those molecules data can be used, past data can be used to detect the efficiency of some other molecules or understand which would work better based on that data. So basically taking the past data and applying machine learning algorithms in pharma industry would help in every aspect of patient care. Now, unsupervised learning is a machine learning technique wherein models are not supervised using a training data set. Okay. This is a machine learning technique where we are not supervising by using a training set. We don't have an output variable in the unsupervised learning. Okay. So if we don't have an output variable, how is it helping? Here, the model themselves find the patterns and the insights from the given data. Okay. It can be compared to learning, which takes place on the human brain while learning new things. So unsupervised learning models will try to find the hidden patterns and insights from the given data. You don't have an output variable, so it will find it by itself. And it is comparable to what our human brain does when we are learning newer things. And the goal is to, however, find the underlying data structure of the data set. Okay. Once we find the underlying structure, we can group that data according to similarities and represent that data in a compressed format. The resultant groups of clusters will have low variance. Okay. Taking the historical data or past data, putting it into machine learning algorithm, and then the algorithm will understand the rules or patterns. Next, we can predict the new data by using these rules or patterns and predict the outcome. Now, how does clustering work? Here we are telling that we don't have output variable or y variable. We are just taking the data x1, x2, x3 and then trying to cluster it. Okay. Now say these are the few unlabeled data. This is one data point, this is one data point, this is one data point, and so on. And now you are applying an unsupervised learning algorithm, that is clustering. Okay. In clustering also there are many types. We are dealing with k-mean clustering. In k-means clustering, the algorithm analyzes the data. Okay. Once the data is sent, the algorithm takes the data and then first it initiates the random centroids. Okay. These random centroids are initiated, and whichever data point is near to these centroids, those data points are put into that cluster. Okay, because these green data points are near to this centroid, it is put in this cluster, and red data points are near to the X centroid, so it is put into this cluster, and it's the same way over here. Now, how these distances are measured? There are various metrics to measure the distances. The one usually followed is the Euclidean distance. Okay. Find the distance between this point and this point, and it will also compare the distance between this centroid and this centroid. So this particular green point is nearer to this X compared to this X. So wherever it is near, it is put into that cluster. So that is how it divides into cluster. But this is not the end. The point is the k-means algorithm runs on multiple iterations. First, it randomly initializes the centroid, assigns the data points. But there are some data points which keep changing. Okay, sometimes this data point is nearer to this centroid, but then next, after calculating and readjusting the centroids, this particular data point might be nearer to the other centroid. So this is classified under this cluster. After classifying this under this cluster, now the average of these data points is taken and a new centroid is marked. This new centroid might be again nearer to this data point compared to this one. So like that, the data points keep shifting from cluster to cluster under every iteration until they achieve a particular point wherein the amount or the distance traveled is minimum. Okay, then the algorithm converges. Now, K-means is one such unsupervised learning method that aims to group similar data points in clusters. So all the similar data points are clustered in one cluster. It allows us to cluster the data into different groups and a convenient way to discover the categories or groups in the unlabeled data set on its own without the need for any training. Okay. And it is also a centroid-based algorithm, as we saw, where each cluster is associated with a centroid. Now, the main aim of the algorithm is to minimize the sum of distances between the data point and their corresponding clusters. As I said, firstly, why are we doing this clustering? We are trying to group similar data points in one cluster. We are telling that these data points are similar, these data points are similar, these data points are similar. But how are we calculating that similarity based on the distance? This distance is being measured by Euclidean distance. So we are telling that these data points are similar, these data points are similar, and these data points are similar. Okay. By understanding the similarity, we are putting them into clusters. This all is being discovered by k-means by itself. Okay, you're not giving any output variable. It is discovering the patterns on its own and putting the data points into the clusters. Okay, so grouping the similar data points. Okay, now the aim of this would be to group the data points in such a way that the distance between the data points is very less. Okay, within sum of squares. So within the cluster, the distance between the data points should be very less. And distance from this data point to the next centroid should be very high. So this would be between sum of square. Okay, this distance between the data points should be less, whereas the distance from other clusters should be very high. That is the mechanism how unsupervised learning works. So step by step, if we want to analyze the k-means, then we can follow the steps. First, the model initialize k-centroids. Okay, once you give the data, okay, then you give the centroids. How many centroids should be there? Say, for example, 8. Okay, so where this 8 should be, which should be the centroid, will be randomly initialized in the first iteration. Okay. Now, initializing centroids K. Next step would be for each sample in the set of samples, assign sample to its closest cluster K based on the Euclidean distance. Now, if this is our data, and let me take some data points. For this cluster, this is the central. For this cluster, this is the central. This is the central. Now, firstly, these centroids are randomly initialized. After initializing that this is a centroid, now the algorithm will calculate the distance of this centroid with the data point. And the centroid and the data point, centroid and the data point. The distance between the centroid and the data point. This is calculated using Euclidean distance. Okay, so whichever data point is near, they are put into this cluster. Now, the distance between these two also will be calculated, but this particular data point would be nearer to this centroid compared to this. So, if this is nearer, this is said to be more similar to this centroid. So this will be put into cluster B. The same way, the distances from the data point to their centroids are calculated, and whichever is nearer, they are put into that cluster. Okay. For each sample in the set of samples, assign sample to its closest cluster K based on the Euclidean distance. Okay. Now, once these data points are assigned, the centroid, okay, will be recalculated. First, it was randomly initialized. Now, once the data points are properly assigned into clusters, now the centroid will be recalculated based on the mean of the sample of the cluster. So all these data points, their mean is taken and the centroid is calculated. So there me. Again, this also me. So there will be new centroids. This will be the new centroid. Okay. No, this will be the new centroid. This will. There might be new centroids forming. This will be by calculating the mean of the data points, right? Now, once the new centroid is calculated, now there might be one particular point. Now this particular point, after the new centroid is calculated, this point might be nearer to this data point compared to this. But this was in the A cluster before. Now this will jump into B cluster in the next iteration. Okay. Again, the center moves a bit and it will become nearer to this cluster and again it will jump. So this jumping process goes on for a while until you give a stopping criteria. Okay. Now, repeat the steps 2 to 3 until a stopping criteria is fulfilled. Next, the variance of each cluster and sum is found out. So these 5 steps will be repeated with different initializations. Maybe firstly, one type of centroid is taken. Then how many different centroids have to be taken? You will give that particular measure, different initialization measure, and based on that, the iterations repeat and clusters will be allocated. Okay, so this steps are repeated until you find that centroid position and until the model converges. Okay. So for stability, the k-means are run multiple times called as iterations. In this different iterations, there will be randomly chosen k-values. So first, what are k-values? K-values would be the number of centroids. Okay, how many clusters you want to have? Say 8, 10, 1. So if you give a range of k-values, then for all these iterations, we can calculate the clusters and see which is giving us the better stability. This particular concept will address the multiple local minima values. Okay, so we can achieve a better accuracy. Sometimes a data point might keep oscillating between 2 clusters. In each of the runs, an algorithm will enter an infinite loop. Okay, it keeps oscillating between one cluster to another cluster until it enters an infinite loop. So to stop this or to avoid this, we go for a particular convergence criteria. Or we are telling that do this iteration only for a few numbers or maximum number of iterations, which we provide. More than that, it can't iterate. So it will not be entering the infinite loop. Next, convergence criteria is to stop when a cluster centroid moves less than a certain percentage of smallest distance between any of the centroids. Okay, you can tell if the centroid is not moving at less than a certain percent, say less than 5%, then you can tell it to converge. Compare between cluster variation to within cluster variation. So within the cluster, what is the variation? And between the clusters, what is the variation? That is compared. And within cluster variation should be less, and between cluster variation should be higher. Typical objective function used is sum of squared errors. So we here use it, sum of squared errors to understand the objective. Now, when can we use this, or what are the advantages? So the k-means clustering algorithm is used to group unlabeled data. We don't have an output variable. At that time, you can use this algorithm to group the data, unlabeled dataset instances, into clusters based on similar attributes. How are we grouping? We are grouping them based on similarity. This similarity is calculated based on the distances. If this particular point is near to this point, then we are telling that it will be similar. Okay, the similarity is based on Euclidean distance. K-means algorithm is most used clustering method as it is inexpensive. Computationally, it is very inexpensive, and it can also scale well with large data. You can use this algorithm when there is large data also. So that is another advantage of using k-means algorithm. Okay. It is an appropriate method when you know how many number of clusters you want from the dataset. And then it is faster also. Speed is another advantage of the k-means algorithm. Limitations would be that you have to choose number of clusters before. Okay, before even giving the algorithm or before even building the model, you have to give the number of clusters, then the model will be initiated. Then if there are outliers in the data set, then it will have a strong influence on the results. That is why when we pre-process the data, then we'll have to remove the outliers. Then initial centroid positions have a strong influence on results. As we saw, initially, these centroids are randomly initiated by the algorithm. So this placement or the initial placement is having a strong influence on the results. So that is one more limitation. K-means doesn't work well with clusters which have varying sizes and densities. If there are different sizes and also huge densities in some categories, then it doesn't work well. So overall, just to recap, if this is your data set, okay, these are the different data points which are marked. Once you send this data into the model, that is k-means algorithm. Okay, this algorithm will initiate the k-centroids. If you have given that there should be 3 clusters, then it will initiate the 3 k-centroids. But where should this centroids be? It initiates them randomly. Okay. It will initiate one centroid here, one centroid here, one centroid here. Okay. After initiating that centroid, Now it will calculate the distance of the data points from the centroid. The distance from here, distance here, distance here. So whichever data points have less distance within sum of squared errors. So this distance is less, that will be put into one cluster. The same way, the data points which are nearer to the centroid, they all will be put into one cluster. Okay. Now, after putting this into one cluster, the centroids which were initiated randomly before, now it is recalculated. We take the mean of all these values and the centroid is recalculated. The recalculated centroid may lie somewhere here. Okay, the centroid may lie somewhere. The centroid may lie somewhere here. After the centroid moves a bit, there might be some data points which were in question. So if this centroid would have belonged to this cluster, after moving the centroid, this data point will be nearer to this centroid. So it will be jumping under this cluster. Okay. After that, again, the centroid are rearranged. Okay. Now taking the mean, the centroid will become this. Again, the data point becomes nearer to this centroid and it jumps here. So this constant oscillation between two clusters due to the rearrangement of centroids will make it fall into infinite loop. Okay, it keeps jumping from this cluster to this cluster, again this cluster, again this cluster. So this enters an infinite loop. To avoid this, we are giving something called as convergence criteria. Convergence criteria is nothing but telling that if the centroid doesn't move more, like say you have given 0.1%. If it doesn't move more than 0.1%, so it has moved only this much, you think. So in this, if it has moved very less, then stop the algorithm or stop the iteration. If it is moving significantly, then continue is what you're telling the model. So giving the convergence criteria, or there is another way where you're telling that this oscillation or iteration should be only maximum of 10. 10 iterations of maximum. So then it will only go for 10 iterations, and it won't go into the infinite loop. So once that is done, all the data points will be put under some clusters, and ultimately when the model converges, we will have centroids which no longer oscillate or which no longer move. Okay, so these centroids ultimately will have less within sum of squared errors, means the distance between the centroid and the data points within the cluster should have less errors, whereas the distance from these to this data point from one cluster to another cluster between sum of squared errors should be high. Ultimately, the goal is to cluster similar records. Okay. The advantages that k-means is faster, but disadvantages is that you have to define how many clusters you want beforehand, before even understanding or before even knowing the data. Then advantages, one advantage is that you can scale this. Okay, this can work well with less data also, and if there is more heavy data, huge data or big data, then you can scale this up. So that is also one advantage of K means clustering. K means clustering would be the number of clusters. So by this we come to the end of K means clustering. Now we will be moving ahead with the code part of the model. Okay, how to build the model and the different steps of CRISPR-Cas9. Okay, so this is the Spider IDE wherein we execute the Python codes. Now, as we discussed, there are 6 phases in CRISPR-Cas9. Okay, we use this methodology to almost solve all the projects. The first phase was go for business understanding. Right? Now, in business understanding, as I said, we define the problem, objective, constraint, and also the success criteria. So, in this example, which we are going to deal with now, we will follow the CRISPR-Cas9 methodology. Each of the 6 phases we will be seeing, and we will also see how we can help or use Pharma analytics and give a solution. Now, K-means algorithm is a unsupervised learning algorithm. Okay, this comes under unsupervised as it does not have an output variable. Now, the problem statement. Okay, the problem statement here is that doctors have need to evaluate a lot of factors before recommending a specific drug for medical diagnosis. Okay, before referring drugs or medicines, doctors have to evaluate a lot of factors. Okay, just give me a minute. So the problem of our business is that doctors are facing or doctors have to evaluate a lot of factors before recommending a drug for a particular diagnosis. So we need to help the doctor. Now we are following the CRISPR-Cas9 process. This has 6 phases. First is business and data understanding. Next is data preparation. Then model building, evaluation, deployment, monitoring, and maintenance. So first, let's go ahead with the business understanding. Here we are defining the objective and the constraint. Okay, the objective here is to maximize the convenience of the drug recommendation. Okay, we want to maximize the convenience of drug recommendation or medicine recommendation. As I already said, objectives and constraints are defined using data optimization terms, that is maximize and minimize. Okay, now we want to maximize the convenience of drug recommendation, and constraint would be to minimize the brain drain. Okay. Now, moving ahead to success criteria. In business success criteria, we try to tell how we want to help the business. Okay, we are mainly writing these in terms of percentages. Now, here we are trying to reduce the application process time from anywhere between 20 to 40%. So, we want to reduce this time being taken by the doctors or the hospitals for the application. So, to reduce this by 20 to 40%, we're trying to apply the model. Then, machine learning success criteria. In machine learning success criteria, we try to define how much accuracy or how much properly we want to build the model. So here, accuracy is mentioned in terms of silhouette coefficient. So to evaluate clustering models, we go for silhouette coefficient. And we here want to achieve a silhouette coefficient of at least 0.5%. That is our machine learning success criteria. Okay. Then there is economic success criteria. Here, we are trying to increase the revenues of the pharmaceutical industry by at least 30% due to the enhanced precision in drug prescription practices. So, in economic success criteria, we want to increase the revenues. So, for every business problem we are trying to solve, we use the CRISPR-MLQ methodology. Firstly, we are defining the objectives and constraints, and then we are defining the success criteria that we are going to achieve by building this model. So the proposed plan is to group the available drugs, allow the understanding the characteristics of each group, facilitating a more precise match between medication and medical conditions, thereby optimizing treatment strategies. Now, after business understanding, we are going for data understanding. In data understanding, there is data collection. Okay. So in data collection, there are two methods that we can take data from the primary data source or the secondary data source. Here, data was taken from the client. Okay. Now, once we are done with business and data understanding, we'll move ahead with the data preparation phase. Okay. For the data preparation phase, we can import the libraries which are required. Okay, there are multiple libraries in Python which will help us use particular functions to solve our problems. Now, we are taking Pandas library as PD. Okay, importing the Pandas library as PD. Pandas library mainly helps in data frame manipulation. Then there is SweetWiz, which is an auto EDA library which will help us perform the exploratory data analysis with just one line of code. Okay, all the first moment business decisions, second moment business decisions, all those can be done just by using one line of code with SweetWiz. There are many other auto EDA libraries which can be used, but here So from sklearn, you can use all these libraries for imputation, for scaling, transforming, and one-hot encoding. Then there is k-means algorithm, which can be applied from sklearn again. Then there are libraries for joblib and pickle. Okay. We can import these libraries so that we can use it when required. Next, I am reading the dataset using the pandas library. pd.read_csv will read the CSV file and store it in the variable df. Okay, this is the data frame. Now, this is the data which we are trying to analyze. Okay, so if we try to understand this data is particularly belonging to US patients, wherein the state which the particular patient record belongs to. Then there is package size. Okay, each pack has how many units? So that is the package size. Then there is product name. This is the name of the medicine. Then there are units reimbursed. So this is mainly insurance data. So how much unit were reimbursed by the company? That is the value over here. Okay. And number of prescriptions is another input variable. Here we are trying to see how many prescriptions had this particular drug, which will give us the popularity ratio, and also how many were reimbursed. Now, there are total amount reimbursed. So if there were 101 prescriptions, how much amount was reimbursed for this particular drug by the company? And there is a particular insurance company, that is Medicaid. So how much of it was reimbursed by Medicaid? Okay. Medicaid is another insurance company. Now, this column is telling us how much amount was reimbursed by the Medicaid insurance, and this is telling us how much amount was reimbursed by other insurance companies. So that is non-Medicaid amount reimbursed. Then there is NDC, which is a unique value for drug in the US. So for each unique drug, there will be an NDC value. So these are different inputs in the data. Okay. Now, as soon as we get the data, first we look at it to understand what the input variables are. Then we try to apply the pre-processing steps and EDA to it. df.info will tell us about the different columns and also the data types involved. Okay. So these are our different columns in the data set. First column is unnamed. Then there is state, package size, product name, units reimbursed, number of prescriptions, total amount reimbursed, Medicaid amount reimbursed, non-Medicaid amount reimbursed, and NDC. Okay. And this is also showing us the data type. Okay. State is of object data type, that is nothing but categorical. Then package size is integer. So data type of each column is also shown when you give the function df.info. In this data set, we are dropping few columns which are unnecessary. Okay. First is the unnamed column. This is the unnamed column we have. This has just the index numbers, but because we already have index numbers, we are dropping that. Next, there is state column. State column, if you see, it has only one value, that is AR. So the variance of this column is 0. And as discussed in the pre-processing steps, any column which has zero variance, we try to remove that because it is not giving us much information. Okay. Next. There is this column, non-Medicaid amount reimbursed. This column has only 0 values and it is also not providing us any information. So these unwanted columns we are dropping. We are removing these columns. So df.drop can remove or drop these columns. So the columns we are removing are unnamed, state, and non-Medicaid amount reimbursed. Axis is equals to 1 means it will remove the columns. If you give axis is equals to 0, it will remove the rows. So our new data frame df1 will have the data set which does not have all these columns. So before our data set had 20,645 rows and 10 columns, but now it is reduced to 20,645 rows and 7 columns. Okay. Next, df1.head will show us the top 5 records in the dataset. So these are the top 5 records, all the columns. And this is just for a quick view of whether the columns have dropped. Okay, here which we have dropped the columns. Next. Next, we are applying exploratory data analysis or descriptive statistics. df1.describe will give us all the descriptive statistics, that is the mean, standard deviation, and also the distribution of the data. So at one glance, we can derive some insights from this. Next, we can apply auto EDA also. Now, the product name has different values. So now here we are trying to understand how many unique values or unique medicines do we have. So df1 of product name.unique has shown that there are multiple unique drugs. Let us see the size of it. So unique.size will give us the count. So there are 2,504 unique medicines. Okay, and dot value underscore counts function will give us how many times each unique drug was repeated. Okay, so lisinopril was repeated around 314 prescriptions. Then amoxicillin was repeated for 218 prescriptions, enalapril for 213 prescriptions, and metformin for 209. So this also gives us insights like which is the most used drug, most frequently used medicines, and how many prescriptions have these kind of drugs. So this is about unique drugs. Next, we are using AutoEDA library, that is SweetWiz. We don't have it, you can just import it, and then SweetWiz.analyze and give the data set. This way you can generate the report. Okay, so just by these 2 lines of code, it will analyze all the EDA steps we have learned. Okay, now coming to package size, you can see there are around 20,645 values in the package size. The maximum value, minimum value, this is the range, the interquartile range, kurtosis, skewness, all these things are given. Okay, this is how the data is distributed also. Then the product name has 2,504 distinct values, and how many times each is repeated is also given. So for all the columns, all the different statistics will be calculated, and whichever is required, we can take into consideration and appropriately process our data. Now we are checking if the data set we have has any missing values. Is_null.sum will show us if there are any missing values or not. So packet size has 0 missing. So all the columns have 0 missing values, means there are no missing values in our data set. Next, we are trying to divide the numeric and non-numeric columns. Okay, so if this is the data and we have 7 columns, all the columns which have a numeric data will go under one data frame, and all the columns which have categorical data will go under another data frame. So we are trying to segregate the data set. Okay. To do that, we are seeing for df1.info. So there's only one object or on one categorical data column, and all others are numerical. Okay. Now, here we are writing a code to segregate the data set. Numeric underscore features is equals to df1.select underscore dtypes. Select underscore dtypes will select only the data types from here which you want to select. So you're telling that it has to exclude object columns. Okay, so only the object columns will be excluded and all other columns will be put under numeric underscore features. So we can see this data frame. All other columns are in numeric underscore features except the product name column. Next, we are segregating categorical features. So here we are including the object data type. So our product name column will come into categorical underscore features. So this is the data frame. Once we have segregated this, next we are creating a pipeline. This pipeline is to apply the pre-processing steps which has to be applied to the data and even to the data when a new data comes in. So we are creating a pipeline. We are giving simple imputer. This simple imputer will impute mean of a column if there are any missing values. Okay, because we have given the strategy as mean, we'll calculate the mean of that particular column, and wherever there are missing values, it will impute it. This is one pre-processing step. And the another one we are applying is scaling. That is min-max scalar. Min-max scalar is scaling the data in the range of 0 to 1. So it will make the data unit free. Okay, and it will be in the range of 0 to 1. Now, we have created a pipeline, num underscore pipeline. This pipeline will have these both techniques, that is imputation and normalization. Next, we are fitting the data into the pipeline and putting it into the processed. Okay. This is the pipeline object create. Once the pipeline is created, we are dumping the file into the job loop. Okay. If we want to apply these steps, then we can take the pipeline and then transform it. Okay. So this is our transformed data. On this data, the missing value imputation was done and also the min-max scalar, wherein all the data is converted to the range of, transformed to the range of 0 to 1. Okay, so the data has become unit free now. Next, we can apply the one-hot encoding techniques for categorical data too. These are the steps. So we can create a pipeline for categorical data, then apply one-hot encoding and store it into the process to do. Okay. If we want to transform the data, then we can take the process to do pipeline and then transform it. Okay. After doing that, we are doing clean underscore describe, and this will show us the mean, standard deviation, and distribution too. Now, pre-processing EDA all is done on our data. The next is to build the model. So the model we are building over here is k-means algorithm. So for k-means, we first have to decide how many clusters we need. Okay, we have to give the k value. So what we are doing is, we are first taking an empty list called TWSs. Then we are giving a range of k values. We are not taking one value for k. To avoid multiple lines, we are taking a range of k values from 2 to 9. Okay. So for each of the k value, we are trying to fit the data and calculate the inertia or within sum of squared errors. Okay. This is overall to build a scree plot. Okay. This is a scree plot which will help us decide how many clusters should be given to the model. Okay. This is total within sum of squared errors, and we are plotting that errors with the different k values, that is from ranging from 2 to 9. Now, this scree plot lets us decide how many clusters to be used for building the model. So the point where there is highest change from one data point to another, that is where you select the number of clusters. Okay, so you can use a scree plot or elbow curve, or we can also use knee locator. Knee locator is also one such method. Now, to use knee locator, we can again define a list, then calculate k in the range of 2 to 9. So in the 2 to 9 clusters, we are initiating random. Init is equals to random means it will randomly initialize the k centroids anywhere. Then the maximum iterations it has to undergo is 30. So how many ever times the centroid changes, the maximum iterations which we are giving is 30. So after 30 iterations, it has to converge or stop. N underscore init is equals to 10. So around 10 different types of initializations it should try. Means in first initialization, if the centroid was here, then next it has to try another initialization. Randomly, whenever it initializes different centroids, it has to calculate this for 10 different initializations and then calculate the error. This is fitted on pharma underscore clean data. And then we are appending the inertia scores into the list. Okay. Once that is in, we are plotting this using the knee locator. Okay. This is again a knee locator graph, which has k values in the range of 2 to 9, and inertia, that is within sum of squares. Wherever there is highest change and lowest within sum of squares, we are trying to take that many number of clusters and build a model. Okay. So for building the k-means clustering, we are taking n clusters is equals to 3. K-means is taken from again sklearn and building the model. Okay. Model.fit of pharma underscore clean. Here you are fitting the data set you have processed. Model.underscore labels will give you the labels. So the whole data set which you have passed, the whole 20,000 records, will be now divided into 3 clusters, as you have given n underscore clusters is equals to 3, as this is showing us least errors. Okay. So one cluster would be labeled 0, another would be labeled 1, and another would be labeled 2. Now, once we have built the model, we want to know or evaluate the model. So for evaluating different models, you have different metrics. Okay, for clusters, we have silhouette coefficient. Okay. So for silhouette coefficient ranges from -1 to +1. Okay. This coefficient tells us how similar an object is to its own cluster compared to the other clusters. Okay. So a score of 1 silhouette coefficient means denotes the best, that the data point is very compact. Okay. If the silhouette coefficient is 0, then it means that the clusters are overlapping. Okay. So calculating silhouette coefficient will give us an idea if the model is well or no. Okay. So our silhouette coefficient is 0.69. Whereas in our ML success criteria, we had given that we want to try to achieve at least 0.5 silhouette score. Then Kalinski-Harabas test is another evaluation technique. Here, if the value is higher, then it means these are the better clusters. Okay. Now, this is the Kalinski-Harabas score. Then there is Davis-Baldwin test, where you are understanding that the lower the score, better the separation between your clusters. So values of Davis-Baldwin index can range from anywhere from 0 to infinity. So we can apply any one evaluation technique and appropriately adjust the parameters. So now we are taking the silhouette score from sklearn and then calculating the silhouette scores for different values of k. So for the range of 2 to 9, we are checking the silhouette score and trying to append it into the list. It's taking time to execute. These are the different silhouette scores for the different number of clusters. So for 4 is 0.7, 3, 0.6, 5, 0.66, 6.65, and so on. So whichever has the better score, we will take that many number of clusters. So our best model would be K is equals to 4 as it has highest silhouette score. So K means we are building the best models with the n underscore cluster as 4. Okay. Now, after defining the model, we are fitting the data. Once we fit the data, we can dump this file in Pickle and then store it in the directory. Now, the best models label would be 0, 1, 2, 3 as we have 4 clusters now. So the whole data would be divided into these different labels. So cluster. This is one cluster, this is one cluster, this is one, and this is another cluster. Now, these are our clusters. So the whole data is divided into these clusters. Now we are concatenating these clusters or the output into the original data frame. Okay, so our original data frame DF is being concatenated for the clusters and stored in DF underscore cluster. So these are the columns which were already present in the data set. Cluster ID is our output column. Okay, so we have divided all this data into these clusters. So in the cluster 1, there are these all drugs. Next, there is cluster 3, cluster 2. Okay, so concatenating the clusters and the original data frame, you can even see the head or the top 5 records. Now we are using the group by method to calculate or aggregate the data. So in cluster 0, these many units were reimbursed. In cluster 1, these many units were reimbursed, and so on. Okay. Once we are done with the model, we can store it into the CSV and our directory. Okay. So overall, what we are trying to achieve over here is take the data, then define what you want to do, business understanding, then collect the data, pre-process it, apply the different EDA techniques, and analyze the data. Once this is done, once you have pre-processed the data, then you can go for model building. Okay. In model building phase, we can apply any algorithm based on the use case and the data. So today we were seeing only about the unsupervised learning model, that is K-means. Unsupervised learning. That is K-means. So this K-means is taking this pharma data, which has around 20,645 records. So all these 20,645 records, you have finally clustered them in 4 clusters based on the similarity. This similarity is again based on the distance. This distance would be Euclidean distance. And your main aim is to find or reduce the within sum of squared errors in the clusters, whereas the distance from the centroid of one cluster to the data point of other cluster should be high. Now, once you have clustered, it will be easier for the doctor to recommend drugs based on the cluster they belong to. Okay. Now, here we have initialized different ranges of k-centroids. Okay, so we are checking the clusters and the scores, that is within sum of squared errors for all these clusters. If we are taking 2 clusters, well, how is the error? 3 clusters, how is the error? 4 clusters, how is the error? And so on. So for all these, we have calculated the inertia and then plotted them into scree plot. Okay. This scree plot is helping us to understand how many clusters should we take. Okay. Should it be 2, 3, 4, and so on? So this is scree plot and number of clusters. Based on this, we can say there is also knee locator, wherein we are giving the same range of k values and calculating. Here we have given some hyperparameters like the max iterations and the init function. Okay, how many initializations should be done in the first go? Whatever inertia values we have got, we are then putting it into a list and then plotting this. This plot is again using matplotlib library, plt.plot. Okay, let me go to the curve. Now, this is the plot. Knee locator. Okay, wherever there is a sharp bend, we are trying to see that particular cluster and decide how many clusters should be appropriate. So using the knee locator, we have decided the cluster and then built the algorithm. First, we have built the algorithm on only 3 clusters based on these knee locator and scree plot, and calculated the silhouette score. Initially, our silhouette score was 0.6. Then, what we try to do is, we try to calculate silhouette score for all these clusters. Okay, not just 3. We are calculating silhouette score for 2, 3, 4, 5, 6, 7, 8. Once we have calculated the silhouette score for all these, 4 clusters gave us better accuracy or better silhouette score. Okay. That is why we took 4 clusters in our best model. Okay. Based on all these analysis, our best model clusters is equals to 4. After building that model, we pickled it and we dumped the file. Now, concatenated the initial data frame with the number of clusters so that it is easier to sort the data. Now, once you have divided all the data into clusters, now doctors would be able to prescribe or recommend drug easily based on the cluster they belong. Okay. So this is the process which is followed basically in the data science model building. So these are the 6 phases. That is business and data understanding, data preparation, model building, evaluation, deployment, and monitoring and maintenance. This was about unsupervised learning. Tomorrow, we will be dealing with the supervised learning model. An example for that supervised learning model would be linear regression. How this linear regression can be used in pharma data and how that would be helpful. Next, we'll also be dealing with the text mining, different pre-processing steps which can be applied in text mine. Okay. So overall, India's pharmaceutical sector is expected to grow exponentially, and the recent trends in analytics also make it a very good combination, and there is a huge growth potential when we have the knowledge of both the analytics and the pharma. This is how you can use a domain knowledge and create significant changes in different fields. This does not applies for pharma, it applies for any domain. So analyzing pharma data would basically help patients and also expedite the drug discovery and development process. So that is all for today, guys. So we'll meet tomorrow with regression and text mining.